Hello everyone, welcome to one more brain support presentation. This one will be about ERP and brain visual analyzer, how to make the ERP. Okay, so I will start sharing my screen with you. Then you can see me on the lateral, and I will come to the slides on our blog. Okay, you all can access these slides just going to brainlatam.com and coming here to knowledge base. Just click here and then ERP processing on analyzer and click here. So all the slides are disponible. Okay, then I will start the presentation. Okay, ERP is evented related potential. So it's a potential that happens always when an event occurs. So one of its characteristics is it is reproducible. It's a waveform that always when the the event occur, it will happen on the brain of someone in the respective area. So it also has a time lock. So it will happen exactly after some time, a defined time after the event. And we can also study this time. It's a characteristic that we can study. So the stimulus can be external or internal, so from outside or inside the body. Um, and you can also see the components of the waveform before the event. So our brain sometimes when the event is happening frequently, we anticipate it, and then our brain already has a reaction before it happens. Yes, and there are three types we are talking here: are the sensory, the motor, and the cognitive. Okay, so here is just an illustration about the ERP. So you can see the the voltage from the brain goes up, down, and with like 100 milliseconds precision. Okay, so I will talk about some examples. One is the P3 or P300 somewhere. It's a really high potential that happens after a stimulus, and it's the most studied because it's the easiest to see when it happens. So I think it has the biggest amplitude because there is more activity, and so after 300 milliseconds is we could say it's when we have the more activity after a stimulus. Okay. Um, Now we are talking about the sensory one. It's come from outside, so it's exogenous. Um, of course, we have to go to sensory areas to get it, like the vision, like auditory, and then when the person hears from the auditory, you can you can see it in the auditory cortex. Are the first components, so they happens really fast. Here is the P100. P100 just 100 seconds after the stimulus, the yes the stimulus, and it has clinical value. So you can study how the brain of someone is, if it is working correctly. I have never studied it, but I think if the person is sensing correctly, and then you can diagnose something. So there are also the motor ERP. It of course it is from motor area, and then when you ask someone to move, you are getting two different potentials from a long period of time. Okay, and the cognitive one is when someone like is thinking about something, and the the signal goes to different areas of the brain, and if you get a big if you get a big view of the signal of the brain, you can see different waves, and then each wave is happening in a different place. The main activity of each wave, each part of the wave, because like the information is traveling inside the brain of someone, because they are like thinking about it, and it's going to different areas. Like when you hear something, it goes to the auditory, then it goes to the prefrontal, then the person try to recognize it and go to another area, and it's traveling inside your brain, even if you are not realizing it. Okay, then I will talk a little bit about a tool that we can use to process this data. Is the analyzer 2. Now it's on the 2.2 version. It's from it's from Brain Products, and in Latin America, the Brain support represent Brain Products. So if you want to see the tool, if you want to use it online, you can talk with us. Okay. So the first thing about the analyzer is you have to create your workspace. Then you have three types of files. The first file is the how data. Then after you record something, you will have the how data. It can has the EEG data and the markers. So all of them are on the how data. And the second one is the history files. So every processing you make on the how data go to history files. What it means? It means you can use the same how data without duplicate it on your computer to a lot of different analyses. Then sometimes the each data is really big, like ActChamp can go to 100,000 hertz. So if you make a 1 hour recording with ActChamp, it will take a long time, and it will be a lot of data. So you don't want to duplicate this data for each processing. It will need a lot of space on your computer or different computers. So it's a solution that Brain Products found to you process to make a lot of different process on the same data without have trouble on your computer. And the third file is the export one. Is after you process something, you can send it out of analyzer as a lot of types. For example, you can analyze just a frequency band for a long period of time, and then just say the median of the frequency band on this time, and create a Excel sheet, and then some to send to uh, oh, I forgot the name, sorry. But you can process this and put on your article and create some graphics about it. Okay. So yes, I already talked about it. Oh, something really good is since you didn't change the how data, if you do something wrong, you don't lose anything. Okay. Now I will show some tools that we can use to process ERP. Just a second. Okay, and these are the tools that we will use following each other to create the ERP. Um, this one is from analyzer. So if you need a new reference, since if you recorded it wrongly, or if you want to do it as another article, you can make it. So you just go here on the analyzer, new referencing, um, go transformation, data set preprocessing, channel preprocessing, and new reference. Then you can select the electrodes you want to create as a new referencing, and you can also use two electrodes at the same time, like the median of them, and create the new processing. Okay. Here is showing how to select it. You select the two mastoids, put here, advance, and now you have the new reference to change your data. Okay. Then something really important is to filter your data. So if on this if I can do it on this presentation, I will try to show you real processing, but I'm having some problems with the internet now, so I'm not sure if I will get it. If I don't, I will do another live, and I already have a video doing it, but I will try to show everything I can from the slides now. So to go to filters, you can go here in that data filtering, the type of filters, and just select the rejection band. If you are in Europe, you can select 50 hertz. If you are in Latin America and US, you can select 60 hertz, and then you eliminate the biggest font source of noise. You can also use others data filters. Okay. Here is a tool to you edit your markers. Um, I will give you the most important tip about it. When you are selecting a marker on your EG data, you have to click Control and then hold it and do whatever you want. So if one day you are doing it and you remember to hold Control, it will be a lot easier. Okay. So to go to edit your markers, just go here, others, edit markers, and then you can see all markers and you can see it on your data and you can change it and move it. But don't forget, you have to hold Control before clicking on it. It's useful because a lot of times when you make your data, you do some mistakes because sometimes the markers are manually, so you have to remove some of them or add some of them or move some of them. So, and also edit the name. So it's important you have the option to edit it. And also, since the markers are on the how data, if you edit it, it doesn't edit the how data, the markers from the how data. So you are just editing it on your processing, on your computer, on your on your files, for example. So uh, if you have to do it a lot of times, the best is you share the first modifications with your lab friends, and then give to each of them, and then everyone start the processing. Okay. The segmentation is really easy. You just go to segment analyzer, segmentation, then you make it or from markers, like you select markers and say, for example, if you have one marker, just one point, you can say, oh, every time there is a point, you select 5 minutes after this point and make a part of the data. So you have a lot of different datas of 5 minutes, and each one starting from a point where you put on your with your markers. You can also select it to do it out for everything, just every 10 seconds. You can overlay it to like every 10 seconds, but the second one starts on the 3 last seconds of the last one. And so like every 7 seconds, you have a 10 seconds recording. Okay. The baseline correction is something really important. I would like to show you guys on the screen, but since I can't, now I will try to make an example with with this illustration, okay? So before the zero, you can see there is a line. The data start here, and after the zero, we have the waveform, okay? Uh, Sometimes when you are getting the data, we the voltage change, and in the voltage you are getting from the brain change because the impedance is a little bit different, because the position of the head is a little bit different. It doesn't influence your data if you are comparing in that time. You can see the waveforms, but if you are compare with a data from 3 hours ago, you have a huge difference. Like one waveform will be here and the other will be here. Like the scale is different. So if you subtract one from the other, you get just a voltage. Then it's the baseline correction. If you get a 10 milliseconds waveform, 3 milliseconds before the zero and 7 milliseconds after the zero, you use the 3 milliseconds before the zero to make a median, and then reduce this median from the 10 milliseconds. So if you have the two waves and use it to both will be in the same spot. Okay? It's easier to show you processing, but I try to explain the best way. Okay, and the last one is the average. If you have a lot of segments, go to average and make a median of all of them. Okay, then I will just return here. Um, Now I will check if I can. Okay, I cannot use the virtual machine now, I think. Yes, I cannot use it now, I think. So I will check if there is any questions on YouTube. If anyone wants to make a question now, I will wait for 1 minute before finish the live. Okay. So thank you very much, guys. This video will be on YouTube. The slides are on YouTube. I will try to schedule a new trainment for next week. I have a travel this week, so I cannot do it earlier. But I already did a trainment on Analyzer, and it's on YouTube. So I will put it on the description of this video, and then I will put it on the blog, so you guys can easily get it wherever you search for it. So if you guys have any questions, you can send an email to profi@brainsupport.com. And thank you very much to participate of the live.